Hi, Science 10. Chapter 7 today. We will be doing 7.1 and 7.2 in one video lesson. They're pretty short. The first one, 7.1, is about kind of the discovery of the cell theory and theories on life. And that's really short. And 7.2 is all about microscopes. And so here we go. Some of these things will be very familiar to you, like the 5 characteristics of living things, that they all need energy and nutrients in some form, and they all make wastes, they all grow, they all reproduce and respond and adapt to their environment. So these are kind of a grade 7 review list of all living things. And over many years, we have scientists who tried to come up with their theories on life and living things. And so we start off with Aristotle around 350 BC, who came up with a very famous theory called abiogenesis, also known as spontaneous generation, where he thought that non-living things could give rise to living things because of observations he made, like rats that ran out of the laundry pile, or maggots coming out of a piece of meat, which he would consider a dead thing. And so, of course, that he also said there was mice coming from potato sacks. Anyway, so he was looking at observations, and because there were no other theories at the time, that stuck around for quite a while. So it wasn't until this 1668 when Francesco Redi decided to do some experiments, and he put this rotting meat in a jar. One was open, one was closed, and he noticed that, in fact, there was maggots that came out of the rotting meat, but only if it was opened. And so he saw that it was likely flies who lay eggs that turned into the maggots. And so once that famous experiment was done, he said that there's probably not such a thing as non-living things giving rise to life. There had to be explanation of life giving life. So to add to that, Louis Pasteur did this swan neck flask experiment, and he had this boiled broth that would have killed microorganisms. He had unboiled broth, which might have microorganisms, and he noticed that if there was a way for microorganisms to get in and make the broth cloudy, they would. If he boiled it, of course, it was too sterile and they didn't live in there. But if he sealed off this area, they could not get in there. So he's just looking at how, pardon me, let's just go here in order. Let's rewind. He sterilized a broth. That was number 1 photo. If there was no access to this for any microorganisms to get in this way, then they didn't go in there and make it cloudy. However, this is number 3. However, if he allowed air in and a way for microorganisms to get in, they did get in and make it cloudy. So he is figuring that there had to be a way for life to get to the locations that it was observed. You can watch the video on that if you like, right there. Okay, next guy. So we just look at historically how one guy's ideas led to the next guy. So Virchow said, you know what, it's not abiogenesis, it's actually biogenesis, which means life can only arise from life. And looking at the guys that came before him, like Pasteur and Redi, he was ready to make that public statement and come up with the that conclusion. And then we had more evidence once the microscope was invented. So Robert Hooke, I see there's an E missing here. Robert Hooke was one who used a very simple microscope to look at the bark of cork, and this is what he saw. And he actually called them cells because they look like cells in the monastery. And so that word stuck. And from then on, cells was what we used for those smallest unit of life. And of course, those were actual cells of the bark of the tree that made up cork. They were empty cells, they were just cell walls, but they were evidence of cells. So as we know them today. Next came Leeuwenhoek, who then made a stronger microscope with 500 times magnification, which is more than the standard microscope we use in class. And he looked at all kinds of things like blood cells and pond water. And he saw things moving around, and he called them animalcules. So he said there's something that is smaller than what we can see with our eye. And here's some videos you might want to take a look at. Schleiden and Schwann took everybody's information and they came up with the cell theory. And this is a cell theory that we still stick with today. And so the components of the cell theory is that all organisms are made of one or more cells. It's the smallest unit of life, and they do come from pre-existing cells. So that is a very important part, that life does give rise to life. Okay, that wraps up 7.1, everybody. And so that's why we're going to go on to 7.2 about microscopes, because that was real short and sweet. When we look at the very simplest microscope that started off our view of the unseen world, it was basically one little lens, like a magnifying glass. And then what we use now are compound microscopes, which have two lenses, of course, one here in the objective and one here in the ocular, so that you can multiply, this is usually 10 times, multiply this ocular magnification with whatever your objective is set to. And some of these compound microscopes go up to 1,000 times magnification. Ours in school, the highest is 10 times 40, and so our highest magnification is 400 times, but there are obviously more powerful ones, but it's just an advantage to have those two lenses working together to magnify. Next kind, electron microscopes, are ones that shoot a beam of electrons and make that image called an electron micrograph, and these can magnify up to 1.2 million times at the point of this being written out of your book. And I'm sure that it's more powerful now because this was done a few years ago. And the transmission electron microscope, TEM, is one that will be able to see through very tiny and thin specimens, because the electrons have to be transmitted right through them. And so they can magnify up to what it says here, 750,000 times. So you've got to get electrons right through. It has to be something really thin. The scanning electron microscope, though, by the way, they can make little slices of things to use the transmission electron microscope on. The scanning electron microscope just sends electrons over the top of something, and you get a look at the surface. What's good about that is they can look at living things and not harm them, but they do not see inside of the object. So here's some scanning electron microscopes that see the outside of a living, these are all little microscopic insects that are freaky to see up close. Okay. The confocal laser scanning microscope is something that can make a three-dimensional image, and with a computer, even though it collects two-dimensional images from different angles. And so those are pretty cool as well. That's a nerve cell. These are, I'm not sure what cells those are. And scanning tunneling microscopes. These, with a probe, transmit electrons on the surface and from the probe to the surface, and then they can look at molecular shapes of some really, really tiny things and make a 3D image of it. And so you can check out more about those as well in these links. You can see with bacteria, with the light microscope that we have in class, not that easy to see. Scanning electron, you can see that they're nicely rod-shaped there, seeing the outside. Transmission, you can kind of see through them. And that's what the confocal would look like. You'd have to stain it to see it there. Okay, looking at sizes of things, we might be able to see up to a hair with our eye. The light microscope is great for cells and bacteria. The electron microscope, anything like virus and further down, DNA, small molecules, atoms, you need the electron microscope for those. And when we look at the parts, most of you are very familiar with the microscope, but the ocular in ours is 10 times magnification. And it usually has a little pointer that as you turn it, you can point things out to your partners. Body tube connected to this body tube here, connected to the revolving nose piece, so you can change which objective you are looking at. The smallest one would be one that is 4 times magnification. Next biggest one 10 times, and the biggest one 40 times. And so when you're figuring out what magnification you're actually looking at the object with, you would multiply 10 times that. So under low power it would be 40 times magnified, under medium it would be 100 times, and under high it would be 400 times magnification. Stage clips keep the slide like sturdy on your stage, and you would always use your coarse adjustment knob first so that you can zoom in on the spot before you use the fine adjustment to focus it a little more. The iris diaphragm will allow a certain correct amount of light in so you can see it very clearly. And then the light source is usually just a bulb that is illuminating it. Unless it's not electric, then there might be a mirror there where the light has to be focused up into the specimen with a mirror, and you carry it with the arm. All right, so you have a microscope that you can label. You know that you'd have to remove the cover and put the slide on the stage. Rotate your nose piece so you use the low power objective first. Use the coarse before you use the fine power, and you can adjust the iris diaphragm to let the right amount of light so you can see the most clear image, and always turn it off and put it back on low power, and that way if the next person uses it, generally if you put low power all the way down, it's in focus on low, but the other medium and high power, you'd have to focus it yourself. Always carry it with one hand on the base, one hand on the arm. That's the right way to care for it. Always use lens paper, and make sure that your image is in the center. A couple terms: resolution is the ability to see fine detail, so this has got high resolution here. Magnification is always when you use the ocular. I was saying those are 10 times whatever the objective is. If it's 40 times, then you're going to be looking at it at 400 times. A cover slip is what is put on top of the slide to protect your specimen and not get any stain or fluid or sample in the objective lens either. Okay, field of view is the diameter of the area that you're seeing. So under low power, you're seeing a bigger field of view. Under high power, you're seeing a smaller field of view. The wet mount would be something where there's a dye, stain, or water that is that the specimen is in, whereas a dry mount, there would be no fluid. Sometimes they might use an adhesive spray though, and sometimes we use staining so that the images are more clearly visible. When you're doing microscope drawings, make sure that there is a nice title with a magnification indicated, and labels are generally done off to the side. The field of view, if you know it, would be put on the bottom. So you label things off to the side and magnification on the bottom, like so. Okay, this is a picture of a broken hair and hair with a root tip, so you can see what that looks like. And when we look at the molecular world, if you flip forward in your notes, have to go to page 24 for this information, and pretty much all of these notes are already in the notes that I have posted. So basically 100 trillion cells are found in the body, and each cell in the nucleus has chromosomes that have your genes in them. And so sections of these would be genes for certain traits. So there might be a gene for your hair color and a gene for how to make the growth hormone in your body. So there's genes for absolutely everything about you in those inside the nucleus of your cell. And you have 46 of these chromosomes in every body cell. So if you look at a gene that's on the chromosome and you unravel it, you can see that it's actually made up of DNA, which to me, I always refer to that as the recipe of how to make all the features about you. And there are 4 components or bases that are in DNA. There's adenine and thymine and cytosine and guanine. And these bases, you probably heard about them in maybe grade 9. But those bases will be unique to you, so that the collection of genes that you have are not like anybody else's. And so basically, if you look at all the guanines in any spot in DNA, it's always bonded to the cytosine, G and C always bond. And wherever you see an adenine, you'll see that the adenine is always bonded to the thymine. Genes would be that particular spot on the chromosome that codes for one characteristic or one trait about you. And gene sequencing is when they're looking at what genes are on a particular chromosome and where the genes are that may be altered and result in genetic disorders, like sickle cell anemia, which is these sickle-shaped blood cells. That's a genetic disorder if that occurs. And damage to genes may be one cause of cancer, at which point those cells start growing uncontrollably. So they study genetics for looking at clues about that as well. The Human Genome Project is looking at coding all of the 46 chromosomes that you have, which are 23 pairs, 23 from your mom, 23 from your dad, and looking at finding out where all the genes are, what they code for on your chromosomes, so they can study things like disorders and treat those disorders. Last little bit here is about viruses and viruses and prions. And so viruses are this weird sort of entity that sort of are not really living and not really dead, because they are not made of cells, but they're made of, they do have genetic material in them. And what they do is they can't really live and reproduce unless they are in a living cell. So if a virus can get into a living cell or a host, then they take over all the cellular machinery of the cell that they have invaded, and that's how they make copies of themselves. So they would land on a cell right here, they would inject their genetic material into that cell and take over that cell and make many, many, many copies of themselves. That's how they function. So viruses can maybe stay in a jar on a counter for 100 years, nothing will happen, take them out, give them host cells to live in, and they can go and infect them. So they're kind of a weird thing. Okay, prions, those are proteins that can be disease causing. Also not actual cells, they are proteins. And so they're just another interesting entity you can read about. When we're culturing cells, that means we're growing them. They may grow many cells to study them and see how they reproduce and how they can maybe cure problems with those cells. And then a little bit on stem cells is that they are like blank state cells that we know the body has in different places, and those stem cells give rise to other cells of the body. And most of the stem cells that are being studied often come from like embryos that maybe are donated, that are not used for in vitro fertilization or something, because in embryos those cells are all stem cells and can be given the right like liquid to live in, and they usually call it a cellular cocktail, and they put those cells in there, and those cells can become any body parts that they need. So they can do a lot of research with embryonic stem cells and use them to grow and repair other body parts. So basically in Canada, it's not legal to do stem cell research on embryos, but in other places it is. We also have stem cells in places like our bone marrow, and in the bone marrow, the cells in there can make all sorts of red and white blood cells and even platelets. So they are considered stem cells, and we now know that there's many other places in the body where stem cells also exist that can form many different kinds of cells. And if you'd like to look up a Bill Nye on stem cells, it's a pretty interesting video. And that wraps up 7.2. And so thanks for hanging in there for 7.1 and 7.2. And do some review on this in your notes, and I will see you back shortly.